Hello, it's Musho Guapo. We're at Unit 2 of AP Biology, Cell Structure and Function. Well, that said, let's go. So, this unit is all about cells and organelles, and our main objective for this unit is to describe the structure/function of subcellular components in organelles. And so, organelles are the different parts of a cell that have specific functions and do specific things within the cell. And so we'll talk about each of those organelles as we go on with this video. So we have to start with the size of a cell. So cells are extremely small, and the reason for that is because cells need to be able to interact with the environment and exchange materials. And it does this the best by having a high surface area to volume ratio. And so if you look at this diagram here, with this large cube and this large cube split up into many smaller cubes, we can see that the volume stays constant, but the surface area increases. And this is good for cells because they try to interact with the environment, and having a higher surface area while maintaining the same volume is crucial to be able to interact and exchange materials with the environment most effectively. So cells, and an important thing to know about cells is that they're compartmentalized, meaning that cells have different parts, so the organelles, and each area or each organelle of the cell performs a different function, and this is true for both eukaryotic and prokaryotic cells. Organelles, just like human organs, carry out processes within the cell, just like how our heart pumps blood throughout our body and our lungs oxygenate that blood. Organelles do functions within the cell, or it does processes within the cell that are vital to the cell's continued. It helps the cell continue to live, and that's what the organelles do. So let's start off with the first organelle, which is the nucleus. So the nucleus is the center of the cell. This is the control center of the cell. It stores genetic information. It has three main parts. It has the nucleolus, the chromatin, and the nuclear porus. The nucleolus is where ribosomes are formed, and this is where RNA and proteins are combined to form ribosomes. The chromatin is a matrix of DNA and proteins, and this is what makes up the chromosomes in the nucleus. And we have nuclear pores, which controls what enters and leaves the nucleus. And then we have the function, and the function of nucleus is to be the control center of the cell, and it stores the genetic information, and it creates ribosomes. Next, we have ribosomes, and ribosomes are composed of RNA proteins, and there are two types of ribosomes. There are free ribosomes and there are bound ribosomes. So free ribosomes are ribosomes that produce proteins that are used within the cell, and these float around within the cell in the cytoplasm. And bound ribosomes are the ribosomes that are attached to the endoplasmic reticulum, the ER, and these make proteins for use outside the cell. And ribosomes exist both in prokaryotic and eukaryotic cells. And to note, ribosomes are the only main organelle that exist within prokaryotic cells, such as bacteria. The function of ribosomes are as to synthesize proteins and to read instructions from DNA to form those proteins. Next, we have endoplasmic reticulum. Endoplasmic reticulum are networks of membranes and sacs throughout the cell. So you can see in this diagram here, there's a network of these membranes. This is one just attached to the nucleus. And there are two types of endoplasmic reticulum. There are the smooth ER and the rough ER. The smooth ER are when there are no ribosomes attached. So we can see here there are no little dots, which represents ribosomes. And then there's the rough ER, which is where the ribosomes are attached to the ER. The functions are slightly different. The rough ER packages proteins and sends those packaged proteins or polypeptides to the Golgi complex to be further folded and created into more complex proteins. And the smooth ER is synthesized lipids, hormones, and makes replacement cellular membranes. Next, we have the Golgi complex. The Golgi complex is made up of flattened membrane sacs known as cisternae, and it has a similar structure to the endoplasmic reticulum, as they're just folded membranes. You can see here it's a bit of folded membrane, the rough ER, and very similar to the Golgi complex. And there are some key differences, though. There's a cis face, which receives vesicles, such as the ones from the ER, and a trans face, which is which ships vesicles to other parts of the cell. So it may receive a small polypeptide, and it might fold it within the complex and then send it out in a vesicle to the transphase. The function of the Golgi complex is to synthesize and package small molecules such as polypeptides, and it also modifies the proteins and produces lysosomes and waste products that are to be removed from the cell. Next, we have lysosomes, which are created by the Golgi complex, and the structure of lysosomes are spherical organelles, and inside the lysosome contains digestive hydrolytic enzymes, which work best in an acidic environment. And so the lysosome is acidic, just like how in our stomach, stomach acid is acidic and works best in an acidic environment. Our stomach is acidic, just like here in the lysosome. The function of a lysosome is to digest different parts, is intercellular digestion, and it also recycles the cell material, and it removes waste from the cell, and it also performs two important functions that you should know, which are phagocytosis and apoptosis. So phagocytosis is cell eating. So basically, when the cell has energy or it sees a foreign substance, what it does is it'll trap that foreign substance in a vesicle or a vacuole, and then the lysosome uses its digestive enzymes to engulf and digest it, and which you can see in picture A. And apoptosis is when the cell doesn't have not a lot of resources anymore, and it's programmed to die when this occurs. The lysosome will have a vesicle. It'll digest a vesicle that contains a mitochondria, and it'll digest that mitochondria, and it'll keep on doing this until there are no more mitochondria left, and this will result in the cell dying because there's no more centers for cellular respiration anymore. Next, we have vacuoles. The main thing to know with the vacuoles are that they are storage. Organelles, and they're basically membrane-bound vesicles that store different things, whether that be food, water, etc. In animal cells, they're very scattered throughout the cytoplasm. However, in plant cells, there's one large central vacuole that stores water, and the main purpose of that large central vacuole is to retain turgor pressure, and turgor pressure is what allows a plant to remain upright, and plants want high turgor pressure, and so having that central vacuole allows that for a high turgor pressure to be within that cell. Next, we have mitochondria, and we all know mitochondria as the powerhouse of the cell, and the reason why it has that nickname is because mitochondria creates energy for the cell. It creates ATP, and and inside the mitochondria there are two membranes, the outer and inner membrane. The inner membrane has folds in it that increases surface area to create ATP, and inside that inner membrane there's a matrix, and the matrix is where the Krebs cycle, or the citric acid cycle, which is the actual process of creating ATP and cellular respiration, that is where it occurs inside the matrix of the mitochondria, which you can see right here as this fluid between the inner membrane, or the cristae inside the mitochondria. Next, we have chloroplasts, and chloroplasts are unique to only plant cells. Plant cells are the only cells that have chloroplasts, and chloroplasts are double membrane, just like mitochondria, and inside and chloroplasts are the site of photosynthesis. And so we all know photosynthesis, plants converting water, carbon dioxide, and sunlight into energy. That energy is glucose, and it does this through two processes, light dependent and light independent. So how it does this is thylakoid discs, which are these little circular things, are then stacked into grana, which are these stacks of thylakoids, and they exist within stroma. And the thylakoids contain chlorophyll, and so the chlorophyll absorbs sunlight, which is important for the light-dependent reaction, and the light-dependent reaction produces ATP and NADPH, or the electron carrier, and then in the light-independent reaction, which is known as a Calvin cycle, that it takes that carbon dioxide. It takes carbon dioxide in this photosynthesis, and it converts it to glucose through carbon dioxide fixation. And so you can see a representation of the chloroplast with its the grana stacks, the stroma, and yeah. So we've talked about all the different organelles, but here's an important theory that you should know about cells and its organelles. And so it mainly applies to eukaryotic cells. So the main idea with this theory is that some organelles, specifically mitochondria and chloroplasts, were once prokaryotic, independent unicellular organisms. Just like how we have bacteria now, these mitochondria and chloroplasts were once prokaryotes like them. And so the idea is that the idea was that mitochondria and chloroplasts were engulfed at some point by ancient ancestors of eukaryotic cells. And so these eukaryotic cells, instead of digesting the mitochondria and chloroplasts, they created a symbiotic relationship with the mitochondria and chloroplasts. And so the evidence that supports this, or you can see in this diagram here, you have a host cell, which is a eukaryotic cell, an ancestor of eukaryotic cells, and we have a prokaryote, which is not a mitochondrion yet, and this eukaryotic cell digests or engulfs, it engulfs that prokaryote, and that prokaryote becomes a mitochondrion as it forms a symbiotic relationship with this eukaryotic host, or the ancestor of the eukaryotic cell. And then later on, it engulfs another prokaryote, the chloroplast, and that chloroplast again makes a symbiotic relationship, and this eukaryote becomes a plant cell, a photosynthetic eukaryote. And the evidence that supports this theory are that both mitochondria and chloroplasts have double membranes. And so in plant cells, eukaryotes have two membranes. There's multiple membranes within a singular cell. And because mitochondria and chloroplasts have that, it suggests that they are more complex and might have existed independently in the past. Also, mitochondria have their own ribosomes and genetic information. So other organelles don't have any of this. They just have the parts and machinery to create and do whatever they need to. But the mitochondria and chloroplasts, they have their own separate ribosomes and genetic information, and so they could have fully existed and reproduced by themselves. And that comes to the third piece of evidence, is that mitochondria and chloroplasts can fully reproduce independently within the cell and don't require aid from the nucleus. So now that we've talked about organelles, we've talked about how organelles interact with the environment, we've talked about the origins of some of the organelles. Let's now go on to membranes. So membranes exist within all cells, at least plasma membranes do. So in a prokaryotic and eukaryotic cells, so plants, fungi, bacteria, animals, there's always a plasma membrane. So this plasma membrane is a phospholipid bilayer. So if we look at this picture here, there is a hydrophilic head and a hydrophobic tail. We've talked about this in the last unit. And basically what this allows is for the cell to interact with the environment, and it uses membrane proteins to allow large substances or polar substances to pass through the membrane, because polar substances won't interact very well with the non-polar tail. So these membrane proteins help polar substances like carbohydrates to pass through the membrane, and that's very important for interacting with the outside environment. So some cells, like plants, fungi, and bacteria, have cell walls, and these cell walls could be made of chitin, cellulose, or other different materials, but they're cell walls and not cell membranes. And so 3 specific examples are plant, fungi, and bacteria. And some have internal membranes. So we talked about mitochondria and chloroplasts having internal membranes, and this usually is only found in eukaryotes. So the structure of membranes are, we talked about it, phospholipid bilayer, which contains proteins. And an important thing to note is that the bilayer actually has unsaturated fatty acids, which can maintain the fluidity in the membrane and allows for that transport of materials. If it was saturated, it would become very stiff and materials really couldn't pass through that membrane. And obviously, we know that the function of membranes is to regulate transport and a lot of different other functions, such as communication and various other things. So we talked a lot about transport in and out of the cell, but there are 2 types of transport that we should know. There's active and passive transport. So passive transport, I'll start with that, is when substances move from an area of high concentration to low concentration to maintain an equilibrium. And so this is the natural type of transport. There is no energy being used. There's a diffusion through a membrane, and a diffusion through a membrane is passive. So an example of being water moving in and out of a cell is tonicity. This is a completely passive process. So if we look at this diagram here, we look at the 3 different types of solutions: hypotonic, isotonic, and hypertonic. Hypotonic means that the concentration of the solute is the concentration of the solute outside the cell is less than inside the cell, and what happens is the water comes into the cell, and this is good for plant cells because they want high turgor pressure, and we talked about how turgor pressure is what allows plants to be upright, allows for the stems to be upright. But in animal cells, because there is no cell wall, that animal cell, its membrane can actually burst, and that's not good for the animal cell. Isotonic solutions is when the solution outside and inside the cell, or like the concentration of the solute inside and outside the cell is the same, and this is most optimal for animal cells. You can see how the shape of the animal cell is normal here, and that's because there's not a high amount of water being taken out or put in that might burst or shrivel the animal cell. However, in plant cells, it's not ideal because we talked about how they want a turgor pressure. It wants to be upright, it wants to be stiff. And if it's isotonic, that stiffness, that turgor pressure goes away. And finally we have hypertonic solutions, and hypertonic solutions basically mean that the concentration of the solute outside the cell is more than inside the cell. And this is not ideal for either plant or animal cells because this just results in the cells becoming shriveled. And you can see that here, this plant cell becomes shriveled, and the plant cell and the animal cell and the plant cell both become shriveled. And this movement of water, this transport of water, is completely passive. There's no energy being put into this system. This is all nature trying to achieve equilibrium. However, now we have to go to the second type of transport, which is active transport. And active transport is when substances move from areas of low concentration to high concentration. So this is using energy. This is using ATP. And a great example of active transport is the sodium-potassium pump in the plasma membrane. So if you look at this diagram here, the cell wants to keep the concentration of sodium and potassium the same. So it wants to keep a low sodium concentration and a high potassium concentration inside the cell, and a low sodium concentration outside, and low potassium concentration outside, and a high sodium concentration outside the cell. And so to do this, to combat the natural equilibrium, it uses ATP in this protein pump, and sodiums are being sent out using ATP, and that results in the outside of the cell being very high concentration of sodium, and using that residual energy, it takes potassium from the outside the cell and brings it inside the cell. And this this entire system keeps the concentrations constant. And this is important because we want constant concentrations inside and outside the cell for certain substances. But this system always needs energy. This always needs ATP to be used in order for this to happen, because this is moving against the concentrate. It's moving from a lower concentration to a higher concentration. So now we've completed Unit 2 of AP Biology. Good job watching through the video so far. Now let's go on to 5 multiple choice questions that are going to test us with all these different concepts. So let's start off with question 1. And question 1 is asking us which structure-function pair is incorrectly matched. So this is giving us a bunch of organelles, and we have to tell which organelle is matched with the wrong function. So vacuoles, it's saying digestion, waste disposal, and storage. We talked about how vacuoles are storage organelles, so we can cross that off. That is correct. Correctly matched. Chloroplasts, photosynthesis. These are in plants. Chloroplasts have chlorophyll. They are vital for photosynthesis. This is correct. We can cross that off. Ribosomes, synthesis of lipids, detoxification of drugs and poisons. Now, if we go back to ribosomes, we can see that their function is protein synthesis, and so the synthesizing of lipids is actually something that the smoothie R does, and so this actually is wrong. So C is wrong. C is the correct answer. And if we go to lysosome, digestion, a breakdown of digested substances, this is what a lysosome does. So C is the correct answer for this question. Question 2, which of the following statements regarding the nucleus is false? So we have to find which of these about the nucleus is false. Cellular respiration. And cellular respiration is something that the mitochondria does, and it's not something that the nucleus does, so we can cross that off, or we can choose that. That is the correct answer. Houses chromosomes, contains chromatin, and surrounded by double membrane are all characteristics or something that the nucleus has. Question 3 is, which of the following correctly describes what is happening in the following diagram? So we have a pipe in a selectively permeable membrane. We have some water, a low concentration of sugar, and a high concentration of sugar. And then we have a higher amount of water and then equal concentrations of sugar. And so because it's a selectively permeable membrane, we can't, it's not going to allow sugar to pass through. So any choice that says diffusion of sugar or osmosis of sugar is wrong. So we can cross off D and cross off A. So now we have to go to B and C. B says osmosis of water from an area of low water concentration to an area of high water concentration. Whenever it talks about concentration, you always talk about the solute, and water isn't the solute in this case. So B is wrong, C is the correct answer. Next, we have question 4. Which of the following is an example of active transport used by the cell to maintain internal connections of internal concentrations of small solutes different from concentrations in the environment? So if we go back to the sodium-potassium pump, it's trying to maintain the concentration of sodium-potassium. It's trying to keep it constant inside and outside the cell. And that's exactly what's being described in this question. So that is why C, sodium potassium pump, is the correct answer. Finally, question 5, white blood cells engulf invading bacterium in a process called phagocytosis, pinocytosis, receptor-mediated endocytosis, and exocytosis. So this question is asking us, phagocytosis. This question is asking us which of the following is describing an engulfing. If you go back to lysosomes, which is what digests things in the cell, we talked about phagocytosis as being cell eating, and an example of that is white blood cells engulfing a bacterium. And so that is exactly what's being described in question 5. And so the answer is A, phagocytosis. Thank you for watching this video. It's been Mucho Guapo. Join the Discord in the link in the description. Thank you for watching.